We are now looking into the applications of PCR, or why PCR is used as a sort of a diagnostic tool. We understand polymerase chain reaction is a very simple technique of amplification of the gene of interest and making million copies of your recombinant DNA. Now, what are all the basic applications of PCR in terms of biology? The first very important application of PCR is we can very easily detect the presence of novel pathogens of virus. Novel pathogens such as a virus, early detection of virus in blood samples. So, PCR has got lot of applications in medical diagnosis. If ever you want to detect an early symptom of a disease like AIDS or an Ebola virus or a newly spreading virus, immediately whether the patient is infected or no, PCR can give you early clues and you can amplify the gene of interest, make million copies of the gene of interest and exactly know how to combat for developing vaccines before it becomes a widespread disease. And next one is, it is used as a sort of a test for infection with HIV. If you take the HIV virus, the easy way to detect early diagnosis of HIV can also be done by means of using the PCR, and it forms a basic research tool in modern biotechnology. It is the first basic tool in modern biotechnology. It can be used for knowing the DNA samples in tigers, elephants, in an endangered species of plant, in a microbe, in a newly discovered gene, or in an endangered plant which could have the last remains of the DNA. If you want to get maybe the sort of a DNA from even from fossils, it can also be used for studying fossil DNA. Imagine you have an extinct plant which is present in the tropical rainforests, and it is one of the kind of plants which is used for the curing of AIDS, and you want to get the sort of a DNA from the plant. Then in a small leaf extract, and primarily if you know how to extrapolate and remove the sort of a DNA from it, and you will get your sort of gene of interest, and you can amplify the gene of interest, and thereby you can preserve the sort of a DNA as in the form of a germplasm. And another very important applications, as implied, it has got applications in forensic science. At the place where there had been a crime, even with relation to, if you want to exactly know who are all the sort of people who have been as a sort of an convict or who have been primarily a sort of a at the place of crime, it's very easily to detect by means of forensic science. So, it has got widespread applications in disease diagnosis. And it has lot of applications in evolutionary genetics. You can exactly trace the evolution of apes, human beings, chimpanzees, plants from water to land, and you can trace the evolution of any particular sort of a plant. And primarily when you use a sort of a PCR, you can exactly even know about new genome sequences. A genome is a term which is used for the total gene content of a particular sort of a cell in an organism. So, total genome sequences can easily be made, which could run to maybe the volume of 2 telephone directories from any particular sort of a plant, animal, human, or a sort of a microbe, and the entire PCR operation can occur within a very, very short time. So we can also, it has got applications in site directed mutagenesis. If you want to identify a gene of interest and knock out a gene of interest, so that you bring about some sort of a change which is termed as mutations, and mutation characters or the mutant characters can be inherited, then it is termed as site directed mutagenesis. And so thereby we can, by PCR, the most important application is you can isolate the gene of interest, and also clone the gene. The meaning of the term clone the gene is, cloning means making multiple copies of your desired gene in biotechnology. So cloning refers to multiple copies of the gene, multiple copies of the gene of interest, and you're trying to amplify the gene. You're trying to amplify the genes. So the main theme of why you use a sort of a PCR in biotechnology is towards isolation of the gene and cloning of the gene for making multiple copies of the gene of interest and amplifying it within the sort of a cell.